Okay, folks, and just for the movie, again, this is Open Lab on Friday, October 25th. Um, um, we're trying to answer questions of folks here in Open Lab and then also do some review topics. So I have to warn folks, this is not exhaustive, right? It's not going to cover everything, but hopefully just some reminders of some important concepts, right? So the first movie we did, folks, was about 25 minutes. It was a review of antibiotics. So we'll try to do these in um, a series of little movies. So this next one we're going to do is we're going to look at metabolism. We're going to look at, in bacteria, we'll look at glycolysis, fermentation, and then we'll look at aerobic respiration. We'll do really quick anaerobic respiration, really quick oxygenic and anoxygenic photosynthesis. And importantly, folks, we're also going to do the nitrogen cycle because there's some important applications of the nitrogen cycle. Okay? So again, let me see if I can move this microphone up closer to the board. I've discovered that unless I'm pretty close to the microphone, when YouTube's does a closed caption, it has a hard time doing closed captions when I'm very far away from the microphone. So I'm going to try to sneak this up here.
If we move the projector forward, you can put it on top.
Oh, that's a great idea. Thank you. Great idea. Okay. Let's see here. As you guys know, I would never make it in Hollywood. If I put it here, though, that'll block your view. It'll be okay? Okay. Alex, are you okay if I put it up here? Closer. Okay. Okay, folks. And again, I'll try to write bigger because I know that's a big problem with the movies is you can't see my writing. So folks, again, what we'll do is we'll start with metabolism. We are not going to go through the intro to metabolism. We're just going to dive right into the metabolic pathways, all right? So folks, remember that when the first cells evolved, what was Earth's atmosphere like? Anaerobic, right? There was no free molecular oxygen. And so there's no free molecular oxygen. And furthermore, we didn't have cytochromes yet, so we couldn't have electron transport chains, right? So the first early cells, they use a primitive form of making ATP called glycolysis. And it literally means sugar lysing. And classically in glycolysis, if a sugar is going to be used, it's like one of the universal sugars is glucose. So we're going to use glucose as our source of high energy electrons. And glucose is 6 carbons. So we'll see one of the events in glycolysis is the 6 carbon glucose will be broken down into 2 3 carbon end products called pyruvate. And in addition, we're going to have a partial oxidation of glucose. So we're going to have a partial oxidation of glucose, meaning we're just going to pull off some of the high energy electrons, not all. We're going to get a little bit of ATP production. So we're going to get a profit, so-called, of 2 ATP per glucose by substrate level phosphorylation. Right? We'll make our 2 pyruvate as end products. Right? That's where the carbon skeleton from glucose ends up. And we'll also make 2 of our reduced NADH. Right? And you want to recall that each NADH carries 2 high energy electrons from what used to be glucose. So folks, if this metabolic pathway takes 10 steps, how many enzymes should it take? 10, right? We don't need to know the names of the enzymes. We don't need to know the names of the intermediates. We just, again, we want to focus on important events. So if I use a series of arrows to represent the 10 enzyme catalyzed reactions, right? So here at the bottom, you guys, our 6 carbon glucose will end up as 2. That's right, 2 pyruvate or pyruvic acid. Pyruvate is just the ionized form. pyruvate is just the ionized form of pyruvic acid. And again, these are 3 carbon molecules. And again, the carbons used to be part of glucose. And the carbons are still carrying high energy electrons from what used to be glucose because we only pulled off some of the electrons. And then, folks, we said that to get it started, what did the cell have to sacrifice to destabilize the glucose in preparation to getting oxidized and broken apart? Yeah, the cell has to sacrifice 2 ATP to phosphorylate the intermediates to destabilize them, getting ready for splitting, right? And at the end of glycolysis, the cell would have made a total of 4 ATP, again by substrate level phosphorylation. But we said the profit, what the cell actually gained, we have to subtract the 2 ATP it used to get glycolysis started. So that means there's a profit, a gain of 2 ATP per glucose in glycolysis. And then we want to make sure we balance. So if we have 4 ATP coming out, I just have to add the equivalent, you guys, of 4 ATP going in. So we already have 2 ATP going in, so I'm just going to add the components of 2 additional ATP. And then the other big event in glycolysis is the partial oxidation of glucose. We're going to pull off 4 high-energy electrons. And remember, we can't throw them into the cytoplasm because they can cause damage. So we're going to pass them to NAD, our electron hydrogen atom carrier. So we're going to end up with 2 of our reduced NADH, okay? All right. So for those first early primitive cells, little prokaryotic cells, there was a lot of preformed organic molecules available. There had been an era of abiotic chemical synthesis where a lot of organic molecules had been made under conditions of primitive Earth. But the little cells had a problem, and the problem is that the cell has a limited amount of the oxidized NAD. So if the cell uses up all of the oxidized NAD, it creates a roadblock right there. And you'll notice that the ATP would be made after that roadblock, right? So if the cell uses up all of the oxidized NAD, glycolysis stops, no more ATP will be used, and then the cell's going to die. So those early cells had to find a way to oxidize this NADH. And the solution was awesome, you guys. So one solution was just take this pyruvate, which is a waste product anyway, and dump the electrons from NADH onto the pyruvate. Again, it's a waste product. The cell wasn't going to use it. So we'll have 2 pyruvate accepting the electrons from our 2 NADH. So this is what the cell was after, was regenerating the NAD. That's going to keep, let me use a different color here. That's going to keep glycolysis operating, right? That's what we need. We need to regenerate an NAD. And then the pyruvate that gets reduced, it gets reduced to lactic acid or lactate. And again, the pyruvate was a waste product, lactic acid is a waste product here, right? So this process where you have a partial oxidation of glucose, this is an anaerobic process, right? There's no O2. No O2 is required. Partial oxidation of glucose. And folks, this is the fancy terminology. We have as a terminal electron acceptor, so we're talking about the electrons that were carried by NADH. Our terminal, our final electron acceptor, is what's called an internal organic molecule. So internal, meaning the pyruvate was made inside the cell. It's an organic molecule, and it's going to be the recipient of those electrons, right? So it's anaerobic. We have an internal organic electron acceptor. And again, this is a partial oxidation of glucose. The little cell has a profit of 2 ATP per glucose, right? So this process, folks, that we've shown here, this is described as lactic acid fermentation. So fermentation was described by Louis Pasteur as life in the absence of air, right? So remember, there's no air, no oxygen involved. And very often the fermentation pathways, there's many different types of fermentation pathways depending on the enzymes an organism has, but very often they're named after one of their end products. So because we made lactic acid, this is called the lactic acid fermentation pathway. And we'd said in lecture, you guys, this is really cool. Our cells remember how to do this. Like if our cells run out of oxygen and they can't carry out aerobic respiration, our cells can switch to lactic acid fermentation for short periods of time just to make that little tiny bit of ATP there. We've talked a lot about lactic acid bacteria, the good bacteria that colonize our mucous membranes. They make acids, inhibit the growth of potential pathogens. The lactic acid bacteria, through making acids, can help preserve our foods in the absence of refrigeration. So lactic acid bacteria inoculated into milk can make fermented milk products like buttermilk, yogurt, kefir. The low pH helps to prevent spoilage microbes from growing. And also with lactic acid fermentation of vegetables, right? This is a way to preserve food. So when we make sauerkraut or kimchi, for example, again, this is a way we can preserve the food by making the acids and inhibiting the spoilage microbes. So this is a really cool process, I think. Yeah. Okay. And then the other fermentation pathway, you guys, we wanted you to know is alcoholic fermentation. And alcoholic fermentation is carried out by Saccharomyces cerevisiae, or brewers or bakers yeast. And you want to remember in alcoholic fermentation, it's a little bit more complicated because in alcoholic fermentation, so we'll go this way, you guys, for alcoholic fermentation. I'll go down here. It's a little bit more complicated in that we have what's called an oxidative decarboxylation. You don't have to remember that, folks. But we'll first tackle the decarboxylation, which is when we're gonna release one of the carbons from each of the pyruvate, so that they get released as carbon dioxide. So this is why in alcoholic fermentation your bread dough rises. It's the CO2 being released. It's also why if you did fermentation of, say, grape juice or germinated grain in a closed bottle, when you open the bottle, you'll have the carbonation release. That's the CO2. And then the intermediate, folks, here is called acid aldehyde. Acid aldehyde. So this is a 2-carbon intermediate. And it's the acid aldehyde then that will accept the electrons from NADH. So you guys, and I apologize because the way I'm doing my cartoon, it's like an LA freeway. So you guys, let's just presume these are the reduced NADH from glycolysis, right? So they'll dump their electrons onto the acid aldehyde. And again, folks, this is what the little cell is after. It's regenerating the NAD to keep glycolysis operating. And then that reduced 2 carbons, those become our ethanol. Right? So that's the ethanol that's produced when we make beer or wine. Yeah. Okay. So again, folks, this is an anaerobic process, partial oxidation of glucose. We make a little bit of ATP by substrate level phosphorylation. And, you know, initially when life first evolved, this was okay. The little microbes had so many preformed organic molecules that it wasn't a problem to be wasteful. But you can imagine that eventually they started using up those preformed organic molecules, so they started having a food shortage, really. And furthermore, you guys, the acids and alcohols produced during fermentation, they're potentially toxic, right? If they accumulate, we know that they can cause denaturation. So there would have been natural selection for microbes that have a more efficient way of making more ATP per glucose with less toxic end products. And folks, do you remember what that cool thing was? That cool process? Yeah, what do we use? What do we use? We use aerobic respiration, right? Yeah. And folks, remember we said that a key event for the evolution of aerobic respiration was the evolution of porphyrin rings because they permitted evolution of bacterial chlorophyll and then chlorophyll A, which permitted evolution of oxygenic photosynthesis. So Earth's atmosphere became aerobic. And furthermore, the porphyrin rings are important parts of cytochromes, which are important parts of electron transport chains. So the porphyrin rings really were crucial in permitting evolution of aerobic respiration. So so I'm going to erase this for right now, okay? And we'll do the redox reaction that we said for sure would be on the short answer section. And it would be the redox reaction for the complete oxidation of glucose using aerobic respiration. And we want to know which of the reactants will be oxidized, which of the reactants will be reduced. So this is our aerobic respiration redox reaction. And again, folks, as microbiologists, when we see respiration, we know there's an ETC, electron transport chain involved, and the word preceding it is telling us what is the terminal or final electron acceptor. So in aerobic respiration, what's the final electron acceptor? Oxygen, yeah, good. So the redox reaction for complete oxidation of glucose using aerobic respiration, we need a source of high energy electrons, and that's the glucose. And then we need our final electron acceptor, and that's molecular oxygen. This line, you guys, represents all the stages of aerobic respiration. This is glycolysis, Krebs prep, Krebs cycle, electron transport chain, making the proton gradient, and then the proton gradient driving ATP synthesis by ATP synthase. And so eventually what will happen is our glucose is going to be oxidized, and it's going to end up, the carbons are going to end up as carbon dioxide, right? And the carbon dioxide, you guys will see, it's going to be made in the Krebs prep and the Krebs cycle, right? And I just realized, you guys, oh my God, I'm so embarrassed. Back when we were talking about the alcoholic fermentation, that process that I labeled oxidative decarboxylation, I shouldn't have called it oxidative decarboxylation. It was just decarboxylation, so I apologize for that mistake. Okay, it won't appear on the exam. And then, folks, our oxygen, when it receives those tired out electrons, it's going to be reduced to what? To water, yeah? And what folks often forget is, why is the cell doing this? Is to release energy, right? Energy. So make sure to include that in your redox reaction. About a third of it will be used to make what? ATP, right? And the theoretical max is 36 to 38 ATP per glucose. In the real world, the cells never reach that high level, but we'll just use it for now. And then 2/3 of the energy will be released as heat. Good. A lot of it's lost as heat. Yeah. Okay. And so, folks, what we want to do now is ask, so which of the reactants is oxidized? So circle the reactant which is oxidized.
Glucose to
Good.
carbon.
There you go.
dioxide.
So when it gets oxidized, we're going to get carbon dioxide. And which of the reactants will be reduced?
Oxygen. To
Oxygen.
water.
Good. So this is reduction. And this is oxidation. Good. Okay. And so, folks, now, and I know this is really intense and maybe the way I do it not that effective, but what I'd like to do is draw a bacterium and show all the stages of aerobic respiration. Okay, so I'll erase this for right now, and we'll do our great big bacterium. And the key event, you guys, in aerobic respiration, the big deal, the big new strategy that was evolved was the electron transport chain using the high energy electrons to form a proton gradient across the membrane, and then the proton gradient driving ATP synthesis. So you guys, I think what I'd like to do is kind of do the cart before the horse. Let's look at the ETC. the proton gradient and the ATP synthase, because that's the key event. And then all the other steps that we kind of get mired down in, it's just showing how the cell slowly rips off the electrons from what used to be glucose to deliver them to the ETC. And a lot of times, like, I would get so hung up on those steps, I was like, no, the main event is the ETC and the proton gradient. So you guys, in our little bacterium then, okay, we'll have this, the outline of our bacterium, you guys, so we could say that this is E. coli, this is aerobic respiration and E. coli. So this will be the cell membrane, right? And again, folks, the really cool thing about respiration is the evolution of the electron transport chain, right? So, and remember, folks, you don't need to know the names of the individual members of the electron transport chain. Do remember there are cytochromes, right? So remember the porphyrin rings had to evolve before the cytochromes could evolve. So just remember there are cytochromes. Most of the members are made of proteins. There's one lipid carrier, but again, you don't have to know the names of the individual ones. So these blobs then are the ETC, electron transport chain. And if we look at function, well, let me ask you all, what is the function of electron transport chain? What is it accomplishing for the cell?
To create a proton gradient.
Exactly, you guys, that's right. And so often that seems to be a hard concept to grasp. So the function of the ETC is to form a proton gradient across the membrane. And protons, you guys remember, they're just hydrogen ions. And it's important that it's across a membrane. That's why the ETC has to be inserted in the membrane, right? Because we need the membrane to pump the protons across to form our gradient. And the other reason it's important the members of the ETC are in a membrane, they have to be in a specific sequence. They can't be just jumbled together. Yeah. So folks, if we just look at proton pumping, this is how the ETC is going to form the gradient. The certain members of the ETC are involved in active transport. They're going to be pumping protons from inside the cell to the outside in the bacterium. So I'll just put like 3 proton pumping stations here, folks. These are the little proton pumping stations. And when the ETC is functioning, we're going to have higher hydrogen ions on the outside of the cell membrane as compared to the inside here. So you guys, which side of the membrane has the higher hydrogen ions? Yeah, the outside has a higher hydrogen ion concentration compared to inside. Yeah, and thus, folks, which side of the membrane would have the ooh, I have to be careful how I word this. Which side of the membrane would have the lowest pH? Okay, so remember,
Inside.
pH and hydrogen ion is inversely related. I always get confused too. So if we have high hydrogen ions, what about the pH? Low pH, right? Right, compared to inside, right? So again, folks, we have a chemical gradient across the membrane. This is called an electrochemical gradient because not only is it a difference in hydrogen ions, but it's a difference in charges here. So it's electrochemical gradient. It's sometimes referred to as the proton motive force because it's going to act as a battery. It's going to act as a source of potential energy. Right? So that hydrogen ion gradient is a source of potential energy. It's going to act like a battery, meaning that it can be used to do work. And the work we're after is ATP synthesis, right? But before we tackle that, you guys, we want to recognize this is active transport, right? So active transport, we know it requires 2 things, right? We're pumping a substance against this concentration gradient from low to high. We know it always requires a membrane protein, which we have in the ETC, but what else does it require?
Energy.
Energy, right? And this is where folks get really confused, right? So what is the energy source that drives proton pumping?
ATP synthase.
Okay, that was so good, and that's so logical, right? But here, our goal is to make ATP. So here we're not going to burn ATP to make, we're not going to burn or spend ATP to make ATP. What's another source of energy? The cell has been ripping off what used to be glucose.
So high energy electrons.
That's it, you guys. So it's the high energy electrons that used to be part of glucose that are the energy source, like the fuel, kind of like, I don't know if it'd be good to call it gasoline. The electrons are the gasoline that are going to run your pumps, right? So you guys, just for right now, let's just show the electrons, these high energy electrons in blue, right? And they're high energy. Initially, they're high energy, right? They've been slowly stripped off what used to be glucose, right? So let's just show them being delivered here. So our electrons are going to be delivered. They start out as high energy electrons. They get passed from one member of the ETC to the next. And what's happening as they're being passed down the chain?
They're losing energy.
That's right. They're releasing energy. And the model we're using, you guys, it's probably from a biophysicist or biochemist. It's probably not sophisticated enough, but I think it'll work. So is that electrons are releasing energy. The ETC harvests some of that energy to do what? Pump the protons, right? So again, you guys maybe think of the electrons either as gasoline or the electrical current that's going to drive proton pumping. Okay, and where did those electrons come from? Way back when, you guys. Where did they come from? Yeah, exactly, right. That's what the glucose was for. Was it has lots of high energy electrons. Right. So those electrons way back when came from glucose, right? Now, who delivered them?
Good. NAD.
Awesome, you guys, excellent. Right, so you remember that just even in glycolysis, when we strip some high energy electrons, we passed it to NAD to make our NADH. We can call it the NAD and FAD are our electron carriers or hydrogen atom carriers, right? I mean, you say like the Uber or Lyft for electrons. So the NADH delivers the high energy electrons to the electron transport chain, and this is awesome, you guys. We regenerate the oxidized form, and that's what will keep all the other processes working. FADH2, folks, delivers electrons a little bit lower, but it too is delivering electrons to the ETC. Good, you guys. All right, so we know where the electrons came from. Where are they headed at the very end here? Now they're tired out, low energy electrons. We can't throw them into the cytoplasm, right? They're going to cause all kinds of damage. Who's waiting for them at the end? Who's our final terminal electron acceptor? Oxygen. That's it, you guys. So it's a messy equation, folks, but we'll just do it anyway. So we're going to take half an oxygen, and we're going to pick up 2 electrons, and we'll pick up 2 hydrogen ions, sorry. And then the final step here, we're going to reduce our oxygen to make what? Water. There you go. Right. Okay, that was a lot, right? That's if you understand that, you've got it. You've got respiration under your belt, right? But we still have to finish up with, why is the cell doing this? The goal is to make ATP, right? So how are we going to make the ATP? What else is in the membrane here?
ATP synthase.
You got it, you guys, ATP synthase. And you'll recall that it's a protein complex. It has 2 functions. And again, folks, this is really oversimplified, but it'll work for us. What's this channel in here for? That's a proton channel, right? So the protons can't diffuse back across the membrane. They can't flow from high to low concentration. They can't get through that hydrophobic area of the phospholipid bilayer, right? Because they've got a charge. So to sneak back across the membrane, they're going to have a protein, right, or a proton channel, and that's one of the functions of ATP synthase. It's a water-filled channel where the protons can flow from high hydrogen ion concentration to low, right? And because again they're charged, think of it almost like as an electrical current. It's causing conformational changes in the ATP synthase. And the second function of ATP synthase, it has active sites to bind ADP and inorganic phosphate, right? As the protons are flowing through, we're getting these conformational changes, we're getting rearrangements of electrons, so that the ATP synthase, the second function is, what is it going to make? ATP, right. It's going to phosphorylate the ADP to make our inorganic phosphate. So you guys, what is the energy source for ATP synthase? The proton, yeah, the flow of the protons, right? So indirectly, the ETC is helping to make the ATP. Indirectly, because the electron transfer chain forms a proton gradient, and then it's the proton gradient, the flow of the protons through ATP synthase that drives ATP synthesis. So many books say the ETC makes ATP, and that's just really misleading, right? ETC makes a proton gradient drives ATP synthesis. Okay, you guys all right with that? Yeah.
So is ATP and the inorganic phosphate, is that on the ATP synthase?
Yeah.
Okay,
So
so
there's
is
a
it actually
binding. Yeah,
connected?
these are active sites, substrate binding sites.
Okay.
Yeah, for ADP and the inorganic phosphate.
Phosphorylate, okay.
Yeah, and then through electron rearrangement, we're going to add that third phosphate to the ADP, and that's what gives us the ATP, adenosine triphosphate. Yeah. Okay. So you guys, if you got that, you got the whole thing, really, because all the rest of it is just the details of how the cell slowly rips off electrons and passes them to NAD and FAD, and in the process tears apart the carbon skeleton, right? And I know for me as a student, I was always so hung up with that stuff. And again, you guys, this is the main event over here. This is the drama, Anyway, so you guys, we'll just do it really quick. So let's say stage 1 is glycolysis, and again, folks, all we're doing is slowly ripping off high energy electrons, right? So you guys, in glycolysis, let's just put the important end products just to keep our life simple, okay? So you guys, in glycolysis, what happens to the glucose skeleton? How does it end up?
It turns into pyruvate.
Awesome. So we get 2-pyruvate. Did we strip off any high energy electrons?
No.
Actually, we did. We're starting to rip them off. And where did they end up? Awesome. 2 NADH. Awesome, you guys. Did we profit any ATP? Good. Yeah. This is per glucose. Sorry, you guys. So we have a profit of 2 ATP per glucose by substrate level phosphorylation. Good. Okay. And then, folks, what I call the Krebs prep. And again, just important end products. So what used to be the carbon skeleton of glucose, it's going to end up in two molecules. So remember, 2 of the carbon get torn off and released as carbon dioxide. Good. Oops. And then the other carbons are going to end up is these little acetyl groups, and they're going to be linked to this carrier molecule called coenzyme A. So we got 2 carbons there, and here's the other 4 carbons of glucose, because we're always tracking where the carbons are going and where the electrons are going. And did we rip off any high energy electrons? Yeah, all these steps are slowly ripping off those high energy electrons. And so what did we make, you guys? Yeah, 2 NADH2. Good. Right. And then what we're going to do is these 2 little acetyl groups, they still have some high energy electrons. So you want to finish ripping off all the high energy electrons. And as a result, we're going to totally tear apart the carbons and release them as CO2. And that's going to be in the Krebs cycle, or TCA cycle, or citric acid cycle. So you guys, do you remember per glucose, the cycle turns twice, right? So per glucose, do you remember how many NADH we're going to make? Excuse me, how many reduced NADH we'll make?
1 turn.
And this will be per glucose, so it'll be 2 turns. It'll turn twice.
6.
Awesome. Oh my God, you guys are good. And what about FADH2?
2.
Awesome. And what's going to happen to what used to be the carbon of glucose?
Carbon dioxide?
Yeah, and how
2
many?
carbon? 2.
So,
Oh,
right,
4, okay.
because we'll do it 2 turns for 1
Okay.
glucose, okay? All right. Will we make any ATP indirectly? Yeah, 2. Good. Right? So, you guys, it's in the Krebs cycle that we finished tearing apart the carbon skeleton that used to be glucose and blowing it off as CO2, and then we finish ripping off all the high energy electrons. Yeah.
Where did you get the FADH from?
Right. So it too is an electron hydrogen atom carrier. The electrons it carries are a little bit lower level energy than the electrons carried by NADH. So it's stripping off those lower energy electrons during the Krebs cycle. So per turn, per one acetyl-CoA, we'll make one FADH2, right? And so we're saying per glucose, the cycle turns twice. So that's why we're doubling them. Yeah. And here you guys, you want to remember, why is the cell doing this? Because it needs to get those electrons delivered where? ETC, right? So these guys are going to go over and donate electrons. These guys go over, donate electrons to the ETC. These guys go over, donate their electrons. And again, FADH2, the electron's a little bit lower energy, so they too will donate the electrons, but a little bit lower in the ETC. Yeah. Okay. So I think that's it, you guys, just for that kind of quick review.
So then ripping off the carbons from skeleton, that's what you call decarboxylation.
Yeah. So if we have a carbon molecule and we start ripping off carbons and releasing as CO2, it's called decarboxylation. Yeah. And so I shouldn't have used that phrase oxidative decarboxylation with alcoholic fermentation. That was just straight decarboxylation. Where we used oxidative decarboxylation was in the Krebs prep and the Krebs cycle.
For oxidative?
Yeah, in oxidative decarboxylation, you're releasing carbons and you're stripping electrons. So you see both CO2 being made, and you're going to transfer some electrons to NAD to make NADH2.
So then what's the difference between that and substrate level phosphorylation?
Okay, so oxidative decarboxylation, stripping off electrons, ripping off carbons and releasing as CO2. Substrate level phosphorylation is a way we can phosphorylate ADP. We can add a phosphate group to make ATP. And the way it happens is like there's some other molecule that has a phosphate group, and in substrate level phosphorylation, that molecule donates its phosphate to ADP to make ATP. This is called oxidative phosphorylation. Yeah, this is called oxidative phosphorylation. And the way I think of it is you have the redox reactions of the ETC forming the proton gradient, and then the proton gradient drives phosphorylation of ADP using ATP synthase.
And that's only found in the ETC.
That is right. Yeah, oxidative phosphorylation, you've got to have an electron transport chain. Yeah, good. Good job. Okay. So, and again, folks, I just want to emphasize, this is not exhaustive, but hopefully just kind of, triggers those memories, right? Because it's been a long time since we've talked about this. So you guys, my thought would be it's 10:30 now. We have another half hour. So let me stop this little movie, right? So we're trying to break it up into smaller pieces. And then, again, folks, let me stop the movie and ask you what you want me to go over, right? Because I know we talked about DNA replication. So since we only have half an hour left, how about you guys tell me what you want to go over? And let me just stop the movie, and we'll and then we'll do another, probably the third little movie, and it'll be number one, the topics you all want to go over.
I was just thinking about that replication board.
Let's do that. I think that's a good idea.
I have many things going in, coming out.
It's very complicated. The DNA replication